Hello everyone, how are you all? Welcome back to the class, guys. I hope you all are doing really great. And now we are moving forward in our plant physiology unit. We have reached to lecture number 2, and this unit will all about photosynthesis, guys. Each and every aspects of photosynthesis we will be seeing in detail. And by the PYQs, if you'll analyze the PYQs, you will see that 80% of the question in all these years, they are focused upon only the process involved in the photosynthesis. That means cyclic, non-cyclic, photophosphorylation, cyclic, non-cyclic, electron transport. So if you see analyze question, 80% of the times they have asked upon this. That means this is the most important thing of this particular photosynthesis, right? So let us start photosynthesis, guys. Till now, what do you photosynthesis? Now we all know that because of this simple process, because of this process is not simple, because now we are seeing the complexity of it in a very easy manner. Don't worry about it. Because of this process, life is surviving or life is there. Now this process is not only it is useful for like two things, right? We all know this. That means food is getting prepared and oxygen, we are getting oxygen, right? So these two things are happening. This everybody knows. Even if you are a non-bio, non-science student, this much we study about, and we are in a competitive phase also. We know this much about photosynthesis. Also, if I ask you that what happens in photosynthesis, simply you will say that photosynthesis, food is getting prepared with the help of light, in the presence of light, and with the help of chlorophyll. This much every person must be knowing, guys, right? Irrespective of the background, right? Now, if I ask you, you must also be knowing this simple equation about this photosynthesis, that photosynthesis, in order to happen, it involves basically carbon dioxide, it involves water, it needed water. Now, carbon dioxide will be taken by the air, atmospheric air. Which part of the plant is participating in this gaseous exchange? Stomatal opening. About stomata, we have understood, right? And in stomata, we have seen that water vapor is also getting released in the form of this transpiration process. And also CO2 will get entered through this particular stomatal opening. This H2O we are getting by, you all know that roots is absorbing H2O. Then one thing is in the presence of my sunlight, guys, this is very important, because sun is the source of all energy, and because of this energy in the form of sunlight, with the help of chlorophyll pigment, this is one pigment which is essential for this photosynthesis to take place. So by utilizing this carbon dioxide, water from the soil, I am getting my food in the form of 6C6H12O6. This is my glucose, guys. This is where we are getting our energy, right? And oxygen, what we breathe in order to survive, and water. This is the equation which everyone must be knowing, because this is pre-10th stuff. Till 10th, everybody studies this in one form or another. This much simple thing everybody must be knowing. Now we have to balance the equation. You don't like just don't remember this equation. Also know this 6H2 CO2 molecule, 12 H2O molecule, then 6O2 and 6H2O. This much you have to remember about this equation, that I need 6 CO2 molecule, 12 H2O molecule, one glucose molecule will be formed, because this is balancing, and you don't have to go into the chemistry of it, that how we have to balance it. Don't go into the chemistry of it. Just see this. Right now, what you are observing, that this basic reaction, this reaction, what is happening in photosynthesis reaction, you are observing that two simple compounds, this CO2 and H2O, is combining together to form a complex compound. This complex sugar glucose is getting formed. So this process of where simpler compounds will combine together to form a complex compound is known as anabolic process. Anabolic. Right. And another one is, if I talk about its counterpart, it's catabolic process. In catabolic process. What happens in catabolic process? A complex compound will break into simpler compounds. This happens in respiration. That is another lecture's topic. Don't worry. But catabolic, you remember. Anabolic process and catabolic process. Anabolic process is where simple compounds like CO2, H2O will merge together, will combine together to form a complex compound, that is glucose. Also, you are analyzing that in this photosynthesis reaction, which is anabolic in nature, this light energy is getting utilized. So when light energy or when heat is getting observed, we call that equation or particular reaction as endothermic in nature. Endothermic, where heat is getting absorbed, and heat is getting absorbed in the form of light over here, and this light energy is getting converted into chemical energy, right? How this is happening, all the details which we will be seeing in this particular lecture, don't worry, but this is the basics of it. Very simple, which even a 10th, like till 10th, who have like those who have studied, like irrespective of the background, this much one must know, right? So this is my equation. It is a photosynthesis reaction is an anabolic process and also an endothermic reaction which takes place. Also, you have to know that this equation is happening, like how this glucose is forming. Glucose is forming because of this H2O or CO2. You will say that this glucose molecule is forming because of this CO2 molecule, and this O2 is forming because of this H2O molecule, right? And this particular equation happens in two ways, which we will be seeing. This light reaction, two reaction will be taking place, guys. Light reaction and dark reaction. We will be studying this in detail. Right now this is not the right time to discuss this, but we have to delve into higher studies now. All right, let's move ahead. Now you know that we are talking about this chlorophyll should be present. With the help of chlorophyll, it will convert my this particular light energy into chemical energy. Now where are these particular pigments, photosynthetic pigments are present, guys? Photosynthetic pigments, and the main pigment is chlorophyll. But a lot of chlorophyll has several types: chlorophyll A, chlorophyll B, chlorophyll C, chlorophyll D. Where are they present? First of all, let us see that, right? Now we are going into the depth of it, and this depth you already know. If I tell you to draw chloroplast structure, can you draw it? Just pause the video and try to draw it, whatever you remember, without seeing your previous notes, right? Try to draw it without even seeing anything, right? I am also drawing. I hope you have done the exercise. This is my, double membrane structure, this chloroplast. You know that over here I have, first of all, grana in the form of these thylakoids. I hope you remember this thylakoids unit. Like this whole channel is like whole this particular is filled up with thylakoids, and this thylakoids, one particular thing is called thylakoid and whole is called grana. This much I hope you must be remembering this, and this is joined by Fred channel. I hope you remember this too. And outer surface part we call it stroma. And in the stroma there is presence of circular DNA, double stranded circular DNA, dsDNA. This is the structure which I'm drawing. There is one more enzyme which is present inside this particular chloroplast, which we called it a rubisco, and this rubisco's role is a very much important. This we will see later on. Just see this what is happening right now. Right now, if I tell you, if I ask you, where is this pigments, photosynthetic pigments, most importantly chlorophyll, where it is where it is present? It is present over here, guys. Just wait. It is present on the thylakoid, right? Thylakoids, this thylakoid. This is present on the thylakoid like this. Now there is not one pigment which is present, that is chlorophyll. No one pigment, but a set of pigments is present. How much pigment you will call? 240 to 230, 230 to 240 around pigments are present, and this is known as a photosynthetic unit. Photosynthetic unit, guys. So this 250, 240 to 230 particular pigments will form my photosynthetic unit, and I'll call it one quantasome. This I have taught you earlier also. So this one dot which I am showing you by blue color now, this one dot is not just one pigment. It is 240 to 230 pigments are present over here, guys. And this particular one unit is known as quantasome, and there are different quantasome which is present on the surface of the thylakoids. I hope this much is clear to you. Now these pigments are basically majorly of few types. Let me show you. P pigments, photosynthetic pigments. See guys, over here you have one most important pigment, that is basically chlorophyll. And chlorophyll is occurring in the particular my chloroplast in the form of different chlorophyll A, chlorophyll B, chlorophyll C, chlorophyll D. But most importantly, we are mainly concerned about chlorophyll A and chlorophyll B, guys. Now, chlorophyll A is present in all green plant. Whatever green plant you are seeing, this chlorophyll A will be present over there. But chlorophyll P is also present in all green plants. But exception is over here that it is not present in BGA. It is not present in diatoms. Right? These are not present. This chlorophyll B is not present in blue-green algae. BGA is blue-green algae, cyanobacteria. And diatoms also it is not present. Chlorophyll B. If you talk about chlorophyll A formula, you must know this. This is you have to learn, guys. C55, and you have to ratify H72, O5, N4, and over here Mg. Now you must have known that this chlorophyll is containing a magnesium molecule, guys. So my, even if you are understanding this, automatically there is some content which is available for you for essential plant nutrients macro CAMGS, right? I hope you are remembering this. You can add over here, and there you must have read one point that there is porphyrin ring, right? So I'll show you the porphyrin ring also, but it is not important. But those who want to learn it, to draw it in the exam, you can draw it definitely, but it is time taking, guys. I'll tell you. Now this chlorophyll B formula is also same, C55. Now this H will be 70 over here. Here is 72. Now H will be 70 over here. O will increase 6, rest will be same, N4 Mg. This formula for my chlorophyll B which is present over here. Now guys, see this particular thing that mm, how do my this chlorophyll A looks like, how, what is the structure of it. Let me just show you briefly, right? So these three triangles you will draw first. I'm telling you also how you will draw the structure, because it is difficult to learn this structure, guys. In the end you will understand it how easily you can draw it. Again try to draw three triangles in the left side. Again try to draw three triangles in the downside. Again try to draw three triangles like this. Just wait, like this. It should be like this. Right, now you check out this. Over here, my N bonding will be there. Nitrogen are 4, right? See over here, N4. So this N4 will be attached like this. Now where will be Mg molecule will be attached? Mg molecule will be attached to it in the center. For here, this will be blocked by some particular double O bond and CH bond. Don't try to remember it. What I'm trying to show you is over here you will find CH2, C double bond O, single bond O, then C20H39. That means C20H39 means chain you will see over here of carbon C till C20, right? So this tail is basically my phytol tail. Just remember that the structure of this my chlorophyll containing phytol tail, and this is my porphyrin ring. You must have heard about it, right? Porphyrin ring. Over here also this is CH double bond CH2. Over here it is CH3. There here is nothing, CH3 again. Like this you will see some CH molecule, but do you have to remember it? I would say avoid it. I'm just showing you that this porphyrin ring, this Mg is essential to develop this porphyrin ring. If Mg molecule, if Mg essential nutrient will not be there, then porphyrin ring formation will also be difficult. So just to connect with you this point which we have understood in essential plant nutrients, when you will be studying, when you must have understood this magnesium concept, you must have heard this term for porphyrin. right? Also, if you want to remember it, just search down the chlorophyll structure once, you will see the same ring. If you want to remember it, those who want to remember it easily, they can remember it by this process how I have told you, right? Three triangle process, by this you can learn it. Now this is my main component chlorophyll, which is present on my thylakoid as a quantosum unit. That means 230 to 240 pigments are present. That means like pigment must be present like this, right? And one pigment is chlorophyll. Chlorophyll A is the most important pigment we have. Other pigment is basically what? Just wait. Just wait, guys. Yes. Now photosynthetic pigments. One you have seen my chlorophyll. Second important one is carotenoids pigment, and this carotenoids pigment contain carotene, you must have heard it, and my xanthophyll pigment. Now this carotene is responsible for orange color, everybody must be knowing this, but carotene function is important. This carotene pigment will protect, will absorb the sunlight first. When sunlight will form, will fall upon this particular pigment quantosome, this carotene will be the first which will be interfering the sunlight, so that this chlorophyll A and chlorophyll B can be saved from the damage, the direct damage of the sunlight which the sunlight can cause, right? So carotene function is also important. So in pigments, you remember these pigments first: chlorophyll, carotenoids, carotene, xanthophyll. These are just two, three examples I have told you. There are 240, I have told you, right? 230 to 240 pigments present. Most importantly, which are taking part in the photosynthesis reaction, is this chlorophyll. And now you have known that we have chlorophyll A and chlorophyll B. We have chlorophyll structure in which magnesium is in the center, which is which is essential for the pore firing ring. I hope this much is good. Right now we will understand that pigments is there. Now there is one photosynthetic system. Photosynthesis, photosynthetic systems inside this particular part. We will see this. Right? First, PS. In short, we will say this PS. One system we will see, right? Now just understand this pigment system, guys. This is not pigment system, but photosynthetic system. Photosynthetic system, where this light will fall. Now I was saying that two things are important: chlorophyll. Now this comes under pigment. This presence is in thylakoid, on the surface of thylakoid. Another one is a light. Now you know that light is here, we are talking about a visible light. I hope you know this electromagnetic spectrum, which I have taught you in agroecology also, right? Electromagnetic spectrum, we have this visible area where, if I ask you the nanometers, like what is the wavelength of this particular visible light, then you will tell me 400 nanometer to 700 nanometer around, right? So one scientist known as scientist name I'm forgetting. I'll tell you Arnold something, right? They experiment that when light waves more than this 680 nanometer, they try to project this particular greater than 680 nanometer light, visible light. They try to fall upon this particular leaf or this leaf surface. They understood, or they find out that the my quantum yield, or you can simply say that my photosynthetic yield, means the product which I obtained was less. That means whatever the food I had to get, whatever the glucose that must be formed, was comparatively low, because I have only entertained greater than 680 nanometer visible light. I hope you are understanding this. Now, if I am including both greater than 680 nanometer light, also visible light which is less than 680 nanometer, that means from 400 to 680, every wavelength light is allowed to fall upon that particular plant. That then they observed that the quantum yield, don't worry about the quantum yield, forget it. Then they found out that the product which they got was high. That means something was turning off, something was not activated when this light system was getting falling on that particular thing, right? When greater than 680 nanometer was getting like this light was falling upon that particular area, some pigment was not activating. So what is that? So these scientists termed this particular photosynthetic system that there are basically PS. There are two PS, PS1 and PS2. I hope you understood. On the basis of what? PS1 will work when light greater than 680 nanometer will fall, and PS2 will work when light less than 680 nanometer is also entertained, less than 680 nanometer, and this is more than 680 nanometer. Right, now let us just see more about it. Now I have told you that these are my thylakoids. This is my grana joined by Fred channel to another grana. If I take these two thylakoids, it will be appearing like this. Now this particular part of above this particular region is known as a non-appraised region. Over here, PS first is found, guys, and in this particular region, this is called as appraised region between the thylakoid. Over here, your PS2 will be found out. These two systems will be found out. Now PS1 is, what is this PS1 containing? How it will appear? This particular PS1 is containing basically chlorophyll B and different types of chlorophyll A. Different types of chlorophyll A means depending upon the wavelength, 670, 680, 685, like this, right? But it has one important component related to this, that is one reaction center. Reaction center is a main center. Don't worry about it right now. Just see this, because we have to utilize this PS1 and PS2 information in the upcoming part also, right? So this reaction center, we will call it P700. That means 700 is the wavelength that it will entertain more than 680 nanometers light. And over here you will see other pigments. That's why there I have said that there are around 240, 230 to 240 in one quantazome, and we are taking one quantazome. So these are different types of chlorophyll A, chlorophyll B, chlorophyll C, right? And light will first fall upon this particular pigment. So this is a shield pigment also, we call them shield pigments. Or you can call them light harvesting pigments also, light harvesting, because over here light will fall first. And this light energy will get transferred from pigment to pigment in the form of electron, which we will see it. And this is my major, which will respond to the light or reaction or protons. This is my reaction center. We call it reaction center. Don't worry about it. We will see it in how this will function, how this photosynthesis will take place. We will understand it in greater detail. But PS1 is found in the non-appraised region of thylakoid, whereas PS2, similar way PS2 will be there, P680 we call it reaction center, and over here you will see different pigments because it is a quantasome. Quantasome is containing 240, 230 pigments, right? So this PS1 is present over here. You will see this is the larger picture of it, guys, right? And over here you will see in appraised region. I hope this much is clear to you. I am just trying to get you familiarized with the words which will be used in this particular, like now we are moving forward, these words will be used, this reaction center, PS1, PS2, photosynthetic system will be used, right? And this particular photosynthetic pigments will be used, and they are responsible in it, right? So we will understand now how this process is happening in the form of light reaction and dark reaction. Now if we'll see the mechanism that is involved in the photosynthesis, now the major things are coming, guys. Mechanism of photosynthesis. Now this mechanism of photosynthesis, you know that this glucose is forming. Now how this glucose is forming, ultimately my food product should form, right? So this usually happens in two processes. We study this in two processes, step 1 and step 2. In step 1, this particular light energy which is coming, the source of light energy is with us. This light energy is being utilized by chlorophyll. And what they are doing by utilizing this light energy? Directly forming glucose? No. They are forming chemical energy, guys. Chemical energy. This light energy is first getting converted into chemical energy. And chemical energy in the form of, now you have to remember this guys, adenosine triphosphate, ATP. You know that this is an energy molecule. Just remember this ATP. No need to remember its origin, from where this ATP word has come, why this ATP. Just remember this ATP molecule will be needed for this photosynthesis reaction. And in step 1, with the help of chlorophyll, light energy will be converted into chemical energy. And chemical energy part is ATP and formation of this NADPH2 molecule. Now full form, don't go into the full form. If you want to see, you can see it. But these two will be formed from this light energy first. How? By the use of this chlorophyll agent. NADPH2 generally is a reducing power which reduces. No need to get into this, but you just understand that one NADPH2 molecule is equal to 3 ATP, guys. I hope you will understand this later on also, but right now what you have to keep in mind that this particular reaction is happening when light is there. So can I call this light dependent reaction? That means whenever there is light, then only this reaction will be happening. So this reaction generally known as light reaction. And we will see how this particular thing is happening in detail. I am just giving you an overview right now. Now this particular chemical energy, that is ATP and NADPH2 molecule which has been formed. Now in this particular reaction, we have to form 18 ATP molecules and 12 NADPH2 molecules. We will form it in when we will see light reaction. But we need 18 ATP molecules and 12 NADPH2 molecules in order to form one glucose unit. Right? So over here, when you will see in step 2, this chemical energy will be converted into glucose with the help of this CO2. Now this CO2 is getting utilized, guys. And this glucose will be formed by this chemical energy. Chemical energy means, now you will always remember, in the case of plants, in the case of photosynthesis, whenever I say chemical energy, ATP and NADPH2 should come into your mind automatically. And NADPH2 is equal to 3 ATP molecules. This much you should know. If you want to know more about NADPH2 ATP, you can search them about it, right? But don't do it, guys. It will only kill your time. And this glucose will be formed. Now, are you seeing any light over here? No. So this reaction is basically light independent. It doesn't matter if light is there or not. This reaction will happen. So this reaction, we call it a dark reaction. Now, some people understand it wrongly that this happens only in dark. No, there is a difference, that this is a light independent. Either there is light or not, it doesn't matter, this reaction will go on, right? So this is a light independent reaction and this is light dependent reaction. In these two processes, we will be completing this photosynthesis, and these two processes are complicated. Generally people perceive it as complicated, but I am telling you, just be with me. This By this method, you will you will be remembering all the things about it, right? Let's start with the light reaction first. Now see light reaction, guys. Just wait. Yes, light reaction. So whenever you will hear this light reaction, what should come to your mind? That light is necessary. This is light dependent. Now, whenever light falls upon any surface, there one thing happens. Electron excite, right? So over here, you just remember the role of electrons, that over here there will be an electron transport system. That means electron will be revolving somewhere where we will be seeing it, but it will be all about how this excitation is involved in the production of this electron, and this how this electron is converting energy and forming this chemical energy in the form of ATP and NADPH2, right? This light reaction consists of two parts. One is basically my cyclic electron transport system. Means, why electron transport system? Because of this light falling upon the particular my leaf surface, my chloroplast, in the thylakoids, that particular generation is happening, electron generation is happening, and this is why now this electron is transporting energy. That's why this is ETS, electron transport system. So this light reaction happens in a cyclic way also, where the electron completes its cycle from where it has originated and it will reach to its place, right? And the second one is non-cyclic ETS. Also, it is known as cyclic phosphorylation, phosphorylation, and non-cyclic phosphorylation. What is photophosphorylation basically? Photophosphorylation. What do you mean by phosphorylation? Phosphorylation means formation of ATP, guys. Phosphorylation means formation of ATP. And photo means in the presence of light. So in the presence of light, this cyclic phosphorylation will take place. In the presence of light, this non-cyclic phosphorylation will be take place. That means ATP will be formed. Because I have told you, you keep two things in mind. Electrons will be involved over here, and because of this electrons, chemical energy will be generated in the form of ATP plus NADPH2. This much you remember. Also, because of this light reaction, water molecule is also broken into to give us this O2. This we will be seeing. Just wait. Just remember this part. Now I told you that how much ATP molecules? 18. How much NADPH? 12. This you have to understand, guys. Rectify it. That 18 ATP I need and 12 NADPH2 I need. Right now, just understand this, right? So let us see this how this cyclic electron transport system, or why this there is non-cyclic electron transportation. It is basically based upon the light, what kind of light is falling, and it is basically because of the photosynthetic systems. Now the role of photosynthetic system will come into picture, which I have told you earlier, in short, right? So in light reaction, under light reaction and under light reaction, we are talking about cyclic photophosphorylation. We are talking about this cyclic picture or cyclic ETS, electron transport system. Now over here, only one photosynthetic system work, guys, or photosystem works, which is that this is PS1. I have told you that I have two PS. Let me draw the PS system, right? I have told you I have one in a photosystem. I have a reaction center, and then I have several other pigments which are present, right? This is my reaction center, and these pigments, the role of these pigments is known as basically antenna. These group of pigments will be known as antenna, right? Why antenna? Because there this pigment is are not absorbing the light or are not exciting the electron. Their major role is whatever light is falling upon them, they transfer their energy. Their energy is getting transferred by these particular pigments to the main reaction center. Why it is reaction center? Because it is not only having this particular pigment, plus this particular area is having enzymes also. Now with the help of this enzyme, reaction will take place, and over here electron will excite. This much you understand, guys, what is happening, right? So I have told you that we have pigments present, photosynthetic pigments. I told you I have pigments in the form of chlorophyll, carotenoids, carotene, xanthophyll. These are major pigments which are present. I have told you that chlorophyll A and chlorophyll B is present, right? Now chlorophyll A is the major part, because reaction center, in the reaction center my chlorophyll A is present, guys, over here, chlorophyll A. And I have told you that this PS1, I call it because over here light more than 700 nanometer, if it falls, then only it will activate. So P700 is also named like this, right? P700 or chlorophyll A700. Rest are what? I have told you that these are chlorophyll A subtypes, like chlorophyll A680, like chlorophyll A600, 500, 550, 570, 590. Now you understand these pigments are transferring energy to the higher wavelength pigments. So suppose this is chlorophyll A550. Now it will only transfer to the higher wavelength pigment. So this will transfer to chlorophyll A570, 590. Over here you will see 600, 620. Likewise, this will be transferring. But when they will transfer the energy to this particular chlorophyll A700 nanometer, over here with the help of this enzyme, this reaction will take place, and because of this reaction, one electron will get excited from here. So this electron will be getting excited, and from here the chain will start, guys. So this particular part is doing the role of antenna over here. First light will be absorbed. Now you just understand, when sunlight is falling, sunlight is not falling that I will fall only at the this particular photosystem one, because I am having more than 700 nano. No, sunlight is having all the rays between 400 to 700 nanometers, right? It will be absorbing. So what I am trying to say is, in cyclic photophosphorylation, sometimes what happens, only this system work, PS1 works, but usually both the system will work, because the next, this other PS2 will be having my reaction center as 680 nanometer. That means light falling below 680 nanometer will be activating this particular photosystem too. So this will be also taking place, and they takes place simultaneously, they will work. But sometimes this also works alone. So I am seeing the reaction if it is working alone in the form of this cyclic photophosphorylation. I hope you are understanding. So second part, in which we will see non-cyclic, in that we will see how these two systems will combinedly work. Then you will get a clear picture. All right, just wait, just understand this cyclic part first. So over here, this light has fallen. This light, because of this light containing energy, this energy has is being transferred by these chlorophyll A pigments, subtype pigments, transferred it, and they are protecting it also, the main reaction center, and they are known as shield pigments. Over here carotene will also be present, right? Chlorophyll B will also be present over here. Carotene will be present, xanthophyll will be present in these pigments, and these are my contosomes, right guys? Just wait. Right, now this will move, and this will, when this energy will be reaching to the reaction center with the help of this enzyme which will be present over here in the reaction center, my electron will be excited. Now this excited electron will go where? It will go to one type of it will go to one of the particular enzyme, or we will call it at a particular site, and this site is known as ferridoxin, guys. You have to remember, and this is known by, in books, FD will be written. Ferridoxin, you have to learn this name, new word. All right, this electron will be reached to this ferridoxin, right? What happens always when this particular electron is absorbed? What happens? That they will be absorbing this energy, and then this electron will again be transferring to somewhere else. That's why it is a chain. Now I am talking about cyclic, that means electron has to come its way to again this reaction center. Understand this, right? So electron is emitted and it has reached to ferridoxin. Some of the modern scientists say that first it goes to ferridoxin reducing substance, FRS, then it goes to ferridoxin. So in some books directly this will be written, or in some books it will first go to FRS, then it will go to ferridoxin. Now after going to ferridoxin, it will go, this electron will again go to where? To my, let me show you. It will go to my cytochrome, guys. These are the sites which are capturing this particular cytochrome B6. Right, and this, after this, and but this electron is, first of all, learn these words. What will happen in this reaction, I'll tell you. Then after this electron, after clearing this site, this electron will go to where? Cytochrome F. Now after this particular cytochrome F, it will go to plastocyanin guide, PC, plastocyanin. This is PC, and this electron will move here. And from plastocyanin, it will go back to this reaction center, so that this chlorophyll A 700 or P700 will become stable. P700 reaction center will become stable again. So what are the words you have learned over here? This ferridoxin, this cytochrome B6. See guys, understand like this only. Don't try to go into the details of what is cytochrome B6. It will only kill your time, because nobody will ask you in the question that explains cytochrome B6. Question will directly come, explain cyclic photophosphorylation. When this, the examiner will only see this diagram, and it will see that have you written this cytochrome B6? That much is enough. Now you can go and read about the cytochrome F, PC, ferridoxin, not needed. Right? Just remember the name. Ferridoxin, cytochrome B6, cytochrome F, then plastocyanin, then this chlorophyll A reaction center, this electron will be returned. Now, but what is happening? My objective was to create chemical energy. That means either ATP should form and NADPH2 should form, right? These two should be forming. Where it is forming actually? So these are forming over here, guys. See, when electron will be transferring from ferredoxin to cytochrome, it will be having some energy. This energy will be utilized in converting this ADP, adenosine diphosphate, to adenosine triphosphate. So one ATP is being generated. I am talking about only one electron right now. If I send two electrons, two ATP will be generated. I'm just giving you an example, right? Just wait. So one ATP will be formed over here. Now in the second, when it will go from cytochrome side to cytochrome F side, again from ADP to ATP will be formed. Right. So over here, what you are seeing, that because of this cyclic photophosphorylation, PPR, just I have written in short, two ATPs I am getting just because of this cyclic PPR photophosphorylation, in which PS first system is activated, and if cyclic phosphorylation is happening, two ATPs will be formed. But actually, two system works simultaneously. So sometimes cyclic chain doesn't form. Non-cyclic chains forms, right? So I'll see non-cyclic, then we will combine it together and we'll see how this 18 ATP I am getting and 12 NADPH2 I am getting. Also how this H2O is getting like destroyed, and it is basically the term is photolysis. But I will use it later on. This H2O is being converted into O2. We will see it, right? This much I hope you understood. What you have to remember in this, you will remember that okay, I am talking about light reaction. You will understand, okay, in light reaction I am talking about cyclic phosphorylation, right? In cyclic phosphorylation, cyclic, as soon as you remember cyclic, PS1 should come into your mind. PS1 means P700 should come into your mind. From here you will release one electron. This electron will go to FD, ferredoxin. This ferredoxin, after this electron will go to my cytochrome B6. In the examination, this will not come into your mind. I am telling you. Cytochrome F, then PC, plastocyanin, then again it will go back. Then only it will be called as cyclic. Over here, ATP will be forming two times from ADP. ADP is ADP is adenosine diphosphate. ATP is adenosine triphosphate, right? So this will be forming directly. Don't go into how it is forming and what is the chemical reaction. Don't go into this, or either it can go to FRS, that is ferredoxin reducing substance. Right, guys? I hope this much you have understood. By it, 2 ATP will be formed, right, by this cyclic reaction. Now we will understand non-cyclic one. Now just see, guys, non-cyclic, non-cyclic photophosphorylation. Non-cyclic one we are seeing. Now in non-cyclic, that means the electron will not be completing its path as we have seen in the cyclic one. So in non-cyclic, both the photosystem will work. P700, that is PS1 photosystem. These are my pigments, other pigments. And P680 nanometer will also work. This is my PS2. Right, means whenever light will be falling on the surface, then P700 will be activated because of the light waves which will be greater than 700 nanometer. It will be activating this. Also, those light will be having wavelengths less than 680 nanometer also. So this will be activating this particular part also. That is photosystem 2. Now these are very, very reactive, guys. This reaction center is very active than this PS first. You know the location of PS1 on the non-appressed region. This is on the appressed region, right? Now you know that whenever these systems will be activated, obviously this energy will be transferred from here, from both. Electron will be excited. Electron will be excited. This much we have understood. Now because of this excitement of electron, this here it was going to ferredoxin, but here it will not go to ferredoxin side. Here new side will be forming over here, that is not forming but present on the plant itself. You have to know the name, plastoquinone, PQ. plastoquinone. Here we have seen plastocyanin, that is different. So over here plastoquinone, you will see it in the PPT also. I have added all these diagrams in the PPT also, just see. So this electron will emerge and it will go to this particular plastoquinone. Now the difference between this photosystem 1 and photosystem 2, I have told you that this particular reaction is very fast. It is taking very fast. So the movement of electron is very fast. So the instability created by the emergence of this electron, the electron has been like arised from this particular reaction center, right? So this reaction will be slow, but this is fast. So over here this electron will reach and complete this particular will stabilize this particular P700 or PS1. But over here, if this electron would have come directly to or complete its cycle, then we were calling it cyclic photophosphorylation. Non-cyclic, why? Because this electron which is arising over here will come over here now. How? By different sites. Now the sites are again same. Cytochrome B6. This electron will come from cytochrome B6. In cyclic, we were seeing that from FD, it will go to cytochrome B6. Then ATP formation was happening, right? But over here, FD to cytochrome B6 we were seeing. But it is not happening in this case because from plastoquinone, electron is coming. So ATP will not be generated. Then we were seeing that it will go to cytochrome F. Over here, we were seeing that ADP is converted into ATP. So ATP formation is happening. Okay. Then cytochrome F to PC, plastocyanin, the electron is coming, then electron will go over here and complete or stabilize this reaction center. So where will this electron will go? One cycle you have seen, this electron will go to my NADP positive. Now the role of NADP positive is coming, guys. NADP positive. This electron will go over here, this molecule or this enzyme, you can say. If you will see the full form of this NADP, that it will be like nicotine amino, something like that, right? Don't go into that. Just remember that NAD positive, it will go over here. Now the things are not complete. This particular reaction center is not stabilized. Over here, electron has been risen. So this system is not yet stabilized. So in order to stabilize this particular system, this system has the power to do one phenomena, that is photolysis, guys. Why it is doing photolysis? In order to get the electron so that it can get satisfy itself. So don't think that this electron will go now over here in this particular, no, this thing doesn't happen, right? You might think that this electron can go over here and it can satisfy this particular or satisfy, not satisfy, but stabilize this reaction center, but it doesn't happen. So this reaction center PS2 basically do the photolysis. What do you mean by photolysis? Photolysis means it breaks the H2O molecule. And how much? 4 H2O molecules are being broken by this photolysis process in the presence of manganese, chlorine. And what will happen by breaking? Well, what will happen when this molecule will be broken down by this P68 or PS2 system? O2 will be formed. Sorry, not O2, but it will get divided into H positive and OH negative, right? But these are 4 molecules, so 4 H positive and 4 OH negative. This reaction is simple, right? Now I hope you remember the reaction of my photosynthetic reaction, that is CO2 plus how much? 6 CO2, 12 H2O. Then I was getting glucose plus 6 O2. I hope this much you were remembering. Right. So from where this 6O2 were coming? This 6O2 is coming now, guys, over here. Keep attention. From here, this photo, because of this photolysis, this 4H2O is broken down into 4 H positive and 4 OH negative. This 4 OH negative is going to form my 1 O2 molecule. And simultaneously, it will be forming 2 H2O molecule. Now you understand where this O2 is coming. This O2 is coming from this H2O by the photolysis, which is happening by the PS2 system. Where is this PS2 system present? In the thylakoid, in the non-appraised part. I hope you are understanding this. And this particular P68 nanometer is nothing but chlorophyll A. Rest all are containing chlorophyll B, chlorophyll, this carotene, this is xanthophyll, right? I hope everything is in connection. So till now I have formed this O2 molecule and 2H2O molecule. Plus 4 electrons are also being formed, right? And these because of this chain or this reaction happened just to get these electrons, so that these electrons can go back and stabilize this particular reaction center, that is PS2. So these 4 electrons will go and stabilize over here. But you are seeing over here that I have only excited 1 electron. Over here you are seeing everywhere 1 electron. So don't worry, just do it 4. That means 4 electrons has been excited. So 4 electrons will be transported over here. Right over here you will see 4 electrons. 4 electrons will go. This much I hope you have understood. Now this one molecule of O2 is released by this 4H2O molecule. But I need how much molecules you need? 6 O2 molecules. For one O2 molecule, 4 H2O molecules have been broken down. So for 6, you need 24 H2O molecules to be broken down. But you are seeing that in this reaction, 2 H2O is being generated also. So ultimately, how many water molecules is utilized? 2 H2O only, because 2 H2O is also generated, na? So net is 2 H2O is being utilized. So to generate 6 O2, you have to utilize 12 H2O. That's why 12 H2O is written over here. Also because of the, for balancing the equation, also 12 H2O will come. But you see in the reaction also, you are seeing 12 H2O will give you 6 O2. So over here to get 6 O2, you need 12 H2. I hope you will understand. Now you are left with this 4H positive. Where will this 4H positive will go? This 4H2O will go over here, guys, and it will combine with my NADP positive 4H positive, right? We have. So this will combine with 4H positive. And what it will form? It will form the required product NADPH2. How many? 2 molecules of NADPH2. So by this non-cyclic reaction, I am getting 2 NADPH2, 1 ATP over here from cytochrome B6 to cytochrome F, and 1 O2 molecule. I hope you are understanding this. So now let us just combine in one screen what was happening, right? Try to recall. We were talking about my light reaction, light independent reaction. In light reaction, we were saying that either cyclic, these are taking simultaneously, cyclic photophosphorylation and non-cyclic photophosphorylation is happening. Photophosphorylation is happening. Also, these are known as, I will tell you, like I have told you, ETS, cycle, non-cyclic ETS and cyclic ETS, electron transport system, because here electron is getting transported. Now these cyclic and non-cyclic are going on simultaneously, like, right? So in this particular system over here, I have P700 nanometer of this particular light. Over here I have P680 nanometer. So in cyclic, what is happening? This electron is going to FD. From FD, this is going to, just wait, cytochrome B6, cytochrome F, then you know this plastocyanin, and this is returning over here. ATP is forming from here, ATP is forming over here. This is my cyclic one, or FRS. You can also, there's some scientists believe that first electron goes to FRS. Everywhere you will mention electron, right? This is my cyclic. So somewhere, sometimes cyclic will be happening. Most of the time, those two reactions simultaneously will be taking. So non-cyclic will be happening. In non-cyclic, you are seeing there will need of plastoquinone. So you are understanding words are important. Plastoquinone. After plastoquinone, this reaction is fast. So it will directly go to this particular cytochrome B6, then cytochrome F, PC, and over here. So either this will be happening or simultaneously both of this happening. Now there is not only one, you can say one chloroplast, right? There are millions of cells are having plant cell, millions of plant cells are having millions of chloroplast. Now this chloroplast will be functioning like this. And ultimately, what you will be getting? From cyclic, you will be getting what? In one cyclic if one cycle, if you revolve about this cyclic phosphorylation, you are getting two ATPs, right? And in non-cyclic photophosphorylation, you are getting two molecules of NADPH2 plus one ATP, right? This is in one cycle. If you are doing just one cycle, now you need, in order to, I have told you in the starting also only that you need 18 molecules of ATP and 12 molecules of NADPH2. So in one cycle of non-cyclic, you are getting 2 NADPH2. So how many? You need 12 NADPH2, right? So multiple, can you go through, like, can you revolve, like, if cycles will happen 6 times, this particular thing I'll be getting, right? 12 NADPH2. So this will be fulfilled. 6 cycles of NC. So multiply it by 6 plus 6 ATP you will get from here. Also in cyclic 2 ATP, 6 ATP you have. Can you multiply it by 6 again, that so that 6 cyclic photophosphorylation will be happening. So it will give you 12 ATPs. So you are getting 12 ATPs from cyclic photophosphorylation, and from non-cyclic you are getting 6 ATPs. Total ATPs 18 ATPs what is required. Next you need 12 NADPH2, which is you are getting in non-cyclic one. Photolysis is also happening. Because of this photolysis only, this NADPH2 was possible, because the 4 H positive ions came from breaking of that H2O molecule. I hope you are understanding. Now you have the required chemical energy to go ahead in the dark reaction. Chemical energy, you have got the required chemical energy, guys. I hope you are understanding this much. Just revisit again. Only need, you have to focus upon how these electrons are traveling and on what sides they are traveling. Just you have to understand this. Don't go into the detail. Even if you search a little bit, if it is taking you 5 minutes for 5 minutes for each and every topic, if you are giving more than this, then it will be wastage of your time, guys. Don't give that much time, right? Now just see, you will read all this, right? Before moving to dark reaction, just see over here, I have given you some material for carotenoids. Now whatever you will read, you will remember it, because we have discussed it. Light dependent reaction, I have told you, this light will come, H2O will be broken down. This pi is nothing but ADP plus pi will give you ATP. This you understand. So ADP is forming ATP by the help of this pi, right? So and ATP and NADPH will be formed because of this particular light reaction. How many ATP? 18 ATP and 12 NADPH. You will not forget it now, right? Also, guys, you have seen this particular like chlorophyll formation. I hope you remember this chlorophyll pigment, that particular structure also. See, some people draw it like in the formation of like this. All right, some people likes to draw like this, but the way I have taught you is also good. You can remember it and in that way also. But if you don't want to remember, then don't. It is not required, right? This is my grana, and over here my photosystem first and photosystem second is present. Photosystem first is giving me cyclic photophosphorylation, and photosystem 2, and when they are working simultaneously, it is non-cyclic photophosphorylation. This particular, do you remember the name of what? Photolysis. Photolysis means breaking down of water molecule in the presence of light in order to get the electron, so that PS2 can get stabilized. I hope you are getting this. This is one diagram which is shown frequently in the NCRTs also, guys. Over here, now you will understand. Over here, PS2, that is P60 is written. Over here, P700. Now you know that electron from here will go to FD. But electron from here will excite and go to PQ. You see the PQ, right? But if both are working simultaneously, then this electron will go to cytochrome B6, and cytochrome, they have written it simultaneously. Then this will go to cytochrome F, and over here what will be generated? ATP will be generated. Then electron will go to plastocyanin. And what is this? This is nothing but my thylakoid upper surface. Now if you will see like this, thylakoid upper surface is seen like this, right? Over here what you are seeing guys, then this is going to my electron is going to ferredoxin over here. NADP positive is over here, right? So this 4H positive will come from the photolysis process, and this will give you NADPH. How many? Two molecules of NADPH2. I hope you're understanding. This is a bit complicated diagram, right? Read this particular part. Now you'll understand everything, guys. Photosystem 1, photosystem 2. This is your what reaction 2H2O and D positive. Not important. Now we will see dark reaction, guys. Dark reaction is interesting plus complicated too. Dark reaction. Now, always remember while dealing with photosynthesis, this equation you have to remember always. CO2, how many molecules? 6. How many molecules of water needed? 12. It will, in the presence of sunlight, now you know the importance of sunlight and chlorophyll. They have done their work. But still, I am not able to get this product. Plus 6O2 plus 6H2O. This equation always keep in mind, guys. In light reaction, we have utilized how many components? We have broke down this H2O in the form in the process of photolysis. We have seen the role of PS what 2 photosystem 2 role, how photolysis is being done, how PS2 is satisfying itself by breaking this by taking the electron from breaking it this particular water molecule. We have seen why sunlight is needed. Sunlight is there, then only this particular this light in the presence of this chlorophyll will be converted into chemical energy. So right now I have chemical energy with me. What I am going to do this with this chemical energy? 6O2 has been formed because of this photolysis also, right? So two things are left basically. My 6CO2 is left and this glucose formation is left, and I have chemical energy by the process of light reaction, right? So this CO2 will be fixed with the help of this chemical energy to this glucose, guys. So in dark reaction is all about fixation of this particular CO2, or you can say that this chemical energy will be converted into glucose with the help of CO2, or you can say CO2 will be converted into glucose with the help of this chemical energy, right? That means CO2 has to be fixed now. And over here you will see that chemical energy, I have 18 ATP with me. That means I have energy with me, right? And Na 12 NADPH2 molecule with me. This much I hope you are understanding, guys. So dark reaction is all about the formation of glucose, and how I will utilize the 18 ATP, we will see it. But how this CO2 is working, this CO2 and this dark reaction is taking place where? I know that I have my, let me show you, leaf, right? This is the leaf. Now at any point of take any cell, suppose this cell you have taken, I am zooming it out. In this particular cell you have chloroplast. Chloroplast is a double membrane organelle, right? You know that you have stroma over here, grana, I am sorry, corrected by Fred channel, and over here you have this stroma. You know that this light reaction was taking place in my over the thylakoids. Light reaction was taking place. So over here, chemical energy was being generated in 18 ATP and 12 NADPH. Now this chemical energy will be utilized where? It will be utilized in the stroma because of the presence of one major enzyme which I have told you. And because of the presence of this enzyme over here, this dark reaction happens in the stroma. And this enzyme is basically which is helping you CO2 to fix is a Rubisco, guys. And this is the most abundant for this particular enzyme. They call it the most abundant enzyme on earth. This Rubisco. This is the statement or introductory statement for this Rubisco enzyme. Rubisco full form, if you want to see, then I will tell you that this is ribulose bisphosphate, bisphosphate carboxylase oxygenase. Not important. But what is the function of this ribisco? This ribisco, because of this ribisco, presence of this ribisco, is affixing this CO2, which how I am getting this CO2? I am getting this CO2 either through stomata, right? Over here from stomata opening, CO2 is being entered over here. Also, I am getting CO2 by the process of respiration, because in every living being, respiration happens. And respiration means, that means exhale of CO2 will happen and inhale of O2, or oxygen will be utilized and CO2 will be given out in the reaction where glucose will be broken down. Respiration is our next topic. We will understand it in next class. But you understand, from two areas CO2 is coming. So always there will be abundance of CO2. But in some cases, sometimes CO2 abundance will not be there. So there will be abundance of O2. So the important or the peculiar feature of this ribisco enzyme is, guys, this ribisco enzyme peculiar feature is that it combines with CO2 also, and it combines with O2 also. So when there will be abundance of CO2, which in normal circumstances abundance of CO2 is there, that's why ribisco is favorable for this particular CO2, and this will give me glucose. How it will give you, we will see it. But what happens sometimes, because of certain reasons, for example, if there is high temperature, right, then what happens, because of the high temperature, plant doesn't want to lose its water vapor, right? So stomata opening get closed. When stomata opening get closed, then CO2 will not be able to enter. That means the concentration of CO2 in the plant parts will be less. That means O2 concentration will relatively increase. So this ribisco has to react with this O2. And when ribisco react with O2, then a wasteful reaction happens, which plant doesn't want it, which we don't want it, because over here I'll also tell it that over here ATP, form ATP is lost, that my energy is getting lost and I'm not getting any glucose. And glucose we need it. This is the product which we need, which plants also need, which humans also need, right? And over here U2, O2 is also getting utilized. That means O2 is needed for humans also. That means this is a wasteful process which goes on in some of the plants. We will see it in detail. I am just telling you an overview of what we are going to see. But my area of concern is this particular part. Right, guys? So now we will see that how this particular CO2 is converted into glucose with the help of chemical energy. This we have to see, guys. So what are we having basically? In the stroma, we are having this ribisco enzyme in the form of RUBP. RUBP, right? In the form of this RUBP, I have this enzyme, ribisco enzyme. Now this RUBP is in the ribulose bisphosphate basically, and 1,5 also we add to this structure, because you don't have to go into this much detail, because RUBP is a 5 carbon molecule. You have to keep this in mind, guys. 5 carbon means that if you will draw the structure of RUBP, then you will see like 1, 2, 3, 4, 5, and over here phosphorus will be attached. So that's why 1, 5 RUBP. But you just remember RUBP right now. A lot of new words will come, lot of new terminology will come. You have to understand it, rectify it as it is, because you cannot do anything about it, right? So this RUPP is there. Now this RUPP will be attached to this CO2, guys. Where will this CO2 come? This CO2 will come to this RUPP because of the presence of ribisco enzyme, right, guys? So this and this will combine, and this will give you a product. First of all, we are just seeing the product which will be getting formed in this particular cycle. And why do we call this cycle? And this cycle is called as C3 cycle basically, right? Calvin cycle also, because of the scientist name Calvin cycle is a, but why C3? Over here we are seeing 5 carbon molecule RUBP. This CO2 is 1 carbon molecule. I hope you know this, right? But these two combine with each other and form PGA, guys. What is PGA? Phosphoglyceric acid. What is this? What is the chemical formula? You don't have to go in this much detail, right? Or you also called it phosphoglycerate. phosphoglycerate. Only name is important, phosphoglycerate. This compound is getting formed. Now another thing which you have to remember, that this compound is a 3 carbon molecule. I hope you are understanding. 3 carbon molecule. 5 carbon molecule, you have to keep this number of carbons in your mind, guys, because this is important. So this is a 3 carbon molecule. 3 carbon molecule means something like its structure will be having 3 carbons. I hope I'm clear. After this 3 carbon molecule, over here this will be utilizing some ATPs. So energy will be utilized. Now this ATP which we have generated in the chemical energy in the light reaction, this will be getting utilized and ADP will be forming. So that means ATP has got now utilized. How many ATPs, we will see it. Now because of the utilization of this ATP, now you also understand, guys, this 3, because of this stable compound which has formed, because RUBP is not stable, CO2 is in inorganic form, in organic form. So CO2 combines with this RUBP and forms a stable compound, that is phosphoglyceric acid, that is also phosphoglycerate. That is a three carbon my 3 carbon compound, right? That's why the names come over here C3 cycle, because over here C3 cycle will begin. So that's why we call some plants as C3 plants, because C3 cycles happens to them, whereas some other plants we call it C4 cycle. We will see it further. Right now you just understand it. PGA will utilize some energy, and energy will be utilized and ADP will be created. Now a new molecule will be created over here, that is by PGA, 1,3 by PGA. Again this 1,3 by PGA is a 3 carbon compound. This compound is having, this molecule is having 3 carbons. Right now I'm just seeing one molecule of CO2, one molecule of RUBP, but it is not happening like that. I'm just getting familiarized, getting you familiarized with these new words, right? Then 1,3 by PGA will be converted into the main compound, guys, because of the utilization of this NADPH to NADPH to NADPH positive, like this. So this utilization, and what does this basically NADPH do? This does a reduction, guys. I hope you know the difference between reduction and oxidation. Let me just tell you in brief what is the oxidation and reduction. Oxidation is addition of oxygen. So in any compound, if oxygen gets added, we call it oxidation. If any compound oxygen gets removed, we call it a reduction. Or if any in any if any reaction hydrogen gets removed, we call it oxidation. Or hydrogen gets added, we call it reduction. Or third important difference between oxidation and reduction. If electrons, you remember this LEO, Leo sounds gur. This is a mnemonics. Loss of electron is happening, then I'll call it oxidation. If gain of electrons is happening, I'll call it reduction. So over here, you will see reduction will be happening of this 1,3 by PGA. Right, and this oxidation and reduction is all again ninth class stuff, guys. Ninth class NCERT, chapter number 1 2 in chemistry. You will read about the difference between oxidation and reduction, right? So this reduction will be happening, and over here PGAL will be forming. Phosphoglyceraldehyde. Phosphoglyceraldehyde. Till now we have not reached to carbon. Where is the glucose which I require? Where is that? CO2 is being utilized, ATP is being utilized, NADPH2 is being utilized. Where is that glucose what I want? PGAL again is a 3 carbon molecule. Nothing else you have to know about this particular compound. Now this 3 carbon compound will give you certain, just wait. From here 4 chain will be going on, and over here fructose will be forming because of this particular PGAL. And because of this fructose, this will be converting into glucose. Now fructose is a 6 carbon molecule. If you see the formulas, for example, and glucose is a 6 carbon molecule. So glucose will be forming over here by the process of fructose, and fructose is being like prepared by this PGL, right? I hope this much you understood, guys. First of all, again I am telling you, be clear with the words. Right now this reaction is not completed. but you have to first keep certain things in mind, that I have to fix it atmospheric CO2. Now CO2 is in inorganic form, but I have to make it an organic form, right? So how will I fix this fixation is happening of CO2 with the help of this ribosco enzyme, and this I have this compound also RUBP with me. 1,5 is why, because this have a chain, and 1,5 is a chemical formula name, right? And it is a 5 carbon molecule. Here is I have one carbon molecule. They will combine each other and PGA will be formed. PGA is phosphoglyceric acid. Then after PGA, this is a stable compound. This is a 3 carbon molecule. That's why we name it a C3 cycle. C3 cycle is very useful, because of that only we are getting food, plants are getting food, and everybody else is getting food because of this C3 cycle. Now this ATP is converted into ATP, that means energy is utilized to form 1,3 by PGA. More stable form will be formed, and because of this, and now it will get reduced to phosphocrystaldehyde. In the presence of this, NADPH2 will be converting into NADPH, right? NADPH. Then this PGL will be giving you fructose and several other compounds. We will see what is happening. But cycle is not yet complete, right? Glucose has been formed. Glucose is my product. Plant will utilize this glucose and store in the form of starch, right? This is understood. But this cycle has to be completed. Then only it will be called as cycle. So these three other carbon PGL compounds will be again forming ribulose and some compound from fructose. They will be forming ribulose. And this ribulose will again be going to this particular RUBP. And this RUBP again is a 5 carbon compound. I hope this much is clear. Now certain things should be added. C3 cycle is all about, if you have to remember the carbon count now. How many carbons are involved? Right, this should be in your mind. Now I am again starting the cycle. Reaction, CO2 molecule, how many utilize? 6 CO2 molecule you utilize, right? So you want over here 6 CO2 molecule. I am by yellow, I am what trying to just wait. So 6 CO2 molecule I need. When I am combining this 6 CO2 molecule, I need RUBP molecule also 6 molecule. So 6 molecule of CO2, 6 molecule of RUBP, which is having one molecule of RUBP having 5 carbons. So how many carbons are having into this compound if you say RUBP? One compound is having, molecule is having 5 carbon, but I have how many molecules? 6. So 30 carbons in this particular thing, and 6 in carbon dioxide. So 6 carbon I am having. So total 36 carbon is entering into the chain. I hope you are getting. Now this 36 carbon molecule are entering in a chain. Now this PGA molecule is formed. Now you know phosphoglyceric acid. Now PGA, you know that this is a 3 carbon chain, 3 carbon molecule. So how many molecules of PGA will be formed because of this particular PGA? And you have 36 carbon in the cycle. So 36 divided by 3. You don't have to do this maths. Ultimately, you have to remember that 12. So you now having in the chain 12 molecules of PGA is formed, not one molecule, guys. So you have 12 molecules of PGA, and each molecule of PGA is having 3 carbons. So total carbons is simple mathematics, 36 carbon in the chain. So these 36 carbon chain, you have to maintain this 36 carbon, guys. Over here till here 36 carbon should be with you in PGL, right? So let us see. Now this 12 PGA will be converted into this particular 1,3 biophosphate. So this 12 molecule should be there, because again bio PGA by PGA is also 3 carbon. So 12 molecule will be converted into 12 bio PGA, and by how much? 1 ATP will be giving me 1 ADP. So 12 molecules are there. So 12 ADP, 12 ATP will be utilized to form 12 ATP. ADP, sorry. So we had 18 ATP. 12 has been utilized. 12 has been utilized, right, over here. So how many you are left with? 6 ATP. Now 6 ATP is also being consumed in the cycle. Just watch it. So now you have 12 molecules of this particular carbon. It will again reduced to PGAL, and PGAL again is a 3 carbon compound. So total 36 carbon you are having. Again PGL should be formed. So 12 molecules of PGA are you left with, right? You are left with, right? But over here, NADPH2 is converted into NADPH. So this is about one molecule. So you have to convert 12 molecules. So 12 NADPH should convert into 12 NADPH, right? So you had in the light reaction 12 NADPH. So and NADPH2. So all the NADPH2 is being utilized. So its work has been over now. Still left with 6 ATP. This 6 ATP is left where it will be utilized. We will see it. So PGAL is formed if 12 molecules of PGAL is formed. Now I have told you that the important one is this PGAL will convert itself into 2 carbon, 2 carbon, 2 carbon and 6 carbon. It will divide like this. So this 2 carbon will be a different compound. This 2 carbon will be a different compound molecule. This 2 carbon will be a different molecule and this 6 carbon will form this fructose. If you will calculate this total, this will be a 12 carbon, or I'm really sorry, this will be a not 2 carbon. I'm really sorry guys. Just correct it. This PGAL will be forming or divide itself into two molecules. So two molecules of PGA over here, two molecules of PGA over here, two molecules of PGA will go somewhere else in some other reaction. But my importance reaction is 6 molecule of this particular PGAL will be giving me my fructose. So if you total this two molecule to, you will get 12 molecules, right? So 12 molecules of PGL are divided into 2, 2, 2, 2, and the last one is 6. My area of concern is 6 or 1, right? Because this is giving me fructose. And how many molecules of fructose I will be getting? This is a 6 carbon molecules. So over here, I'll be having 3 carbon, like fructose is a 6 carbon molecule and I'll be having over 3 molecule of fructose over here. So from here, 1 molecule of fructose will be giving you glucose, guys. So 1 molecule of glucose is being formed, only 1 molecule of glucose. So rest of the particular fructose, that is two molecules of fructose are left, right? Two molecules of fructose are left. They will go back. This two molecule of PGB will come back. This will come back, and over here these all molecules will come back. And there are also several reactions happen. I am not confusing it with you because that is not needed, guys. So directly they will try to form this ribulose, and this ribulose is again a 5 carbon thing. 5 carbon ribulose is a 5 carbon compound, or you can say molecule. Here, then after ribulose will be converted into this RUBP by utilizing the 6 ATP we are left with. So this 6 ATP will convert into 6 ADP because over here you have 6 molecule of this particular ribulose, and this will be giving you 6 molecule of RUBP back. So if you will analyze it, now you have again 30 carbon over here, and 6 carbon will again you will take from CO2. Where is this 30? See, you started 36 carbon chain, right? 30, you started with 36 carbon. Till here everything was fine. 36 carbon, if you calculate, 36 carbon are there. But over here in this fructose to glucose, 6 carbons has been utilized to form this glucose. The rest of the 30 carbons are back to the chain so that they can repeat the cycle. I hope I have made this complicated thing in a bit easier way, but it is like you have to understand in this way, guys. There is no shortcut for this. You have to learn. Once more I am just writing the names. First of all, you should know the names, right? Or first of all, you should have this equation in mind. CO2 plus H2O is giving you C6H12O6 plus O2. This much you just remember, and you know that 6 CO2 molecule you need, 12 H2O and 6 O2. 6 CO2 molecules you should have in your hand, right? And over here you know that one glucose should be forming. So you now know that 6 CO2 I have. Now this 6 CO2 should combine with one enzyme, that is RUBP15. So 6 molecule of RUBP I am having. This will go ahead and PGA will be forming. PGA molecules, how many molecules I am having? 12 molecules, because PGA is a 3 carbon compound. Then it is forming 1,3 by PGA. And why? And over here ATP will be lost, 12 ATP will be lost, like into ADP. Then this by PGA will be reduced to form PGAL. Over here 12 NADPH2 will be utilized. Now this PGAL is again having, I have 12 molecule. Out of this 12 molecule, 6 molecule will go back to the cycle, and this 6 molecule over here will give me fructose. So 3 molecules of fructose will be formed. 2 molecules will go back, and 1 molecule will give me this glucose. Now this all will be combining together and will be forming ribulose. How many ribulose? 6 molecules of ribulose will be forming. And this will again utilizing the ATP. How many ATP? 6 ATP will be utilized, and again this will convert into RUBP. So this glucose, one glucose I am getting. Again you will repeat the cycle, second glucose I will getting. This cycle repeats itself. I hope this is clear. Just remember these things, right? So now you see the reaction. Now you will understand all this reaction where you have to focus in any content. Now you see any content, you will be able to understand, guys. Don't see this structure and all, right? They are just confusing. So you have a ribulose 5 phosphate or RUBP 1,5. Remember anything? Sorry, over here you see ribulose 1,5 bisphosphate, RUBP. So carbon dioxide is attached over here. See carbon dioxide. You know now 6 molecules of carbon dioxide is used. So I need 6 molecules of. Because of the Rubisco enzyme, this is because Rubisco in the presence of carbon dioxide behaves as carboxylase, right? So it will go ahead and it will form phosphoglycerate or phosphoglyceric acid. Just see over here. This phosphoglyceric acid will be changed into 1,3 bisphosphoglycerate or 1,3 by PGA. And over here what happened? ATP has been utilized. How many now? You know 12 ATPs because 12 molecules are revolving in the cycle. Then over here NADPS2 will be utilized, you know, and this will convert into NADP positive. Then you see PGAL will formed. So this is glyceraldehyde, phosphoglyceraldehyde, PGAL. It is not written in this particular flowchart, but I'll show you. So 3, and it will go back, and over here your glucose will be forming, guys. So 3 things are happening basically. One is carbon is getting fixed first when carbon dioxide is attached, then reduction is happening because of this ATP and NADPH2. Then there will be a regeneration of a ribulose, and then again the reaction will go on. See this Kelvin cycle. Kelvin cycle because Melvin Kelvin has given the name of this or has find out this particular reaction, right? This is also one reaction which you have to keep in mind. The CO2 will come over here. You have RUBP molecule, ribulose bisphosphate. This will go, this will form a 1,3, this will form PGAL, and this PGAL will go back to the cycle. This one is also a good diagram, guys. See it. Now you will understand. See, everything will be same. Over here, this is CO2. They have also mentioned the carbon count. 1 carbon count, 5 carbon count, 3 carbon count. So you remember what I have taught you and just see the spellings and all from here, right? As it is, you have to remember it. And read this content. Now you will get everything, right? But now it is not completed. The C3 cycle goes on when CO2 is there. But I told you that this Ribisco enzyme is a dicy enzyme. Dicy means it can it has two phases. If CO2 concentration is more, it will react with CO2. It will fix CO2 and carboxylase. That's why it has carboxylase in its name, right? And it will perform C3 cycle, which is very beneficial. Glucose we are getting. But if O2 presence is there, then this Rubisco also combines with O2. And over here, this wasteful process happens. Now, this is not needed to us, guys. This is not what we need, right? So what happens when O2 combines with Rubisco, we will see it. And how some plants have like adopted some mechanism or they have evolved a process where Rubisco and oxygen does not combine. They have evolved their mechanism, or you can say the structure has evolved in a certain way that O2 cannot combine with this Rubisco. We will see this because this is a wasteful process. So some plants have evolved over the period of time to not let this reaction happens. So we will see it. Now my second thing to understand is how Rubisco reacts with O2 and what wasteful product it's formed, right? This we have to understand. Now what happens when CO2 concentration is less and my RUBP has come in contact with this particular O2, guys. Now in this process, what happens that 3 organelles comes together. Just wait, guys. So Just wait, guys. Yes. So I have this Ribisco enzyme with me, RUBP. CO2 concentration is not there. O2 concentration gets high because of the stomatal closing. O2 concentration is high now. O2 will react with it, right? Now, because of this reaction, one new product is formed. In the C3 cycle, one product was forming, right? In this particular, when O2 and RUBP reacts, two products form. Over here, phosphoglycolate is formed, guys, plus PGA, phosphoglyceric acid, which used to form in C3 cycle also, it is get solved. It also formed. Now, this particular PGA will go to the C3 cycle like the previous one we have seen. But what we have to do with phosphoglycolate? This phosphoglycolate is a two carbon molecule. It is containing two carbon molecule. PGA is three carbon molecule. Over RUBP is five carbon molecule. That's why if you will see two carbon, three carbon, five carbon. O2 is not having any carbon, right? So this phosphoglycolate is formed. Now it is a two carbon stable compounds. That why sometimes this particular cycle is known as uh, s- what do we call is C2 cycle, right? Sometime it is called like this. One more name is that I'll tell you what is the name later on. But what happens is this formation is happening. This is happening in one organelle, that is my chloroplast. This is happening in chloroplast. Now the C2 cycle does not remain in chloroplast only. It involves two more organelles, that is one perioxysome, spelling just check guys, perioxysome, and third organelle is mitochondria. Now this is a wasteful product. Plants doesn't want, we doesn't want it to happen. Why? I'll tell you. Now this phosphoglycolate will go and in perioxysome it will convert into some compound. Name you don't have to remember, but if I'll tell you, then it is like glyco glycooxylate, something like that. Then this glycooxylate will convert into glycine. You don't have to remember all this. I'm telling you. Now this glycine will go to mitochondria and some compound will be forming over here. Now because of this glycine present over here, what happens that over here CO2 is released, guys. Are you understanding? In plant has to utilize CO2. In C3 cycle, you were seeing how CO2 has been utilized. Six molecules of CO2 was utilized. But over here in C2 cycle, CO2 is getting lost. So CO2 is an asset for plants. Are you understanding? So this is a wasteful reaction. Why? Because CO2 is getting lost. And where, in what process CO2 get lost? When we respire, right? When respiration happens, CO2 we release out, right? So in the plants also, when respiration happens, CO2 gets released. That's why this process is also known as what? Respiration, not respiration, photorespiration, because this is happening in the presence of light. So photorespiration is the name of this particular cycle, and this is a cycle this has to complete. So this will go back and some product will be formed over here, then this will come over here, and over here ATP will be utilized also. So currency will be utilized, ATP or my energy will be utilized, and the product is nothing, but I am losing this CO2 also. So double loss we are facing over here, and this will again convert to RUBP, and this cycle will again happen. So are you understanding why photo respiration is not good for plants? Or it is not good for us too, because over here we are not getting any glucose compound in the form of food. I'm not getting any like over here you will see that O2 is getting absorbed, so O2 we need, but O2 is getting engaged in the reaction. The CO2 is getting outside, that means CO2 is getting in the form of respiration. That's why it is photorespiration. Energy is getting utilized, so it is a complete wasteful process, guys. So some plants over the period of time, for example, sugar plant, maize plant, they have one certain evolution of this structure. Their structure is such that I'll show you how that they have evolved over the period of time, that this C2 cycle of photorespiration will not happen to them. Why? What is the change in structure? I'll discuss right now. But these type of plants are known as C4 plants. C4. I'll tell you guys. All right. So let me show you this particular, first of all, evolution, what has happened, guys. Let me draw First of all, a leaf. Suppose this is my leaf, right? Over here, you can see a leaf. Now I'm showing you a cross section of this. I have like cut this leaf down over here, and I'm showing you the cross section, a bigger view of this particular leaf. Just check this out. This upper portion I'm trying to show you. It will be something like that, right? Or if I can like make it a bit bigger, right? This upper part of cross section, if I'm cutting them down, there's a 3D view. Try to view it. So you know that over here, first of all, I have some cells. Then I have some openings, and that is basically stomatal opening, right? So I'll be having on the surface of the leaves, I'll be having some stomatal openings, right? And these are my mesophyll cells. What kind of cells are over here? Mesophyll cells. Similarly from the downside also. Now you see that these mesophyll cells in the normal plants, these mesophyll cell are containing my chloroplast. And in normal plants, that is C3 plants, this chloroplast is having rubisco. When this rubisco is present, then this is behaving or this is participating in C3 cycle. Basically, dark reaction is happening. Now, because of the concentration, high concentration of O2 due to high temperature, high light concentration, my O2 concentration has increased nearby this particular stomatal opening, also inside the cell also. That means if now my rubisco is reacting with O2 because stomata has closed, CO2 is not coming. So I have to stop this cycle, right? So over the period of time in sugarcane and maize, one type of evolution has been happened. That means formation of new kinds of cell we can see in these plants over the period of time, like this. And these particular cells are known as bundle sheath cell, guys. And these are formed in some specific plants only. Bundle sheath cells. Over here, you were seeing these cells are mesophyll cells. The unique thing about this bundle sheath cell is that these plants have developed, the sugarcane and maize have developed that this rubisco which was present in the chloroplast of mesophyll cell, which is very easily reactive with the oxygen. Now this rubisco has been taken down to this bundle sheath cell where it is protected. So this oxygen will not be easily interacted or will never get interacted with this particular O2, right? So to get into the cycle of photorespiration. So because of the presence of my this particular ribosco enzyme inside bundle sheath cell, inside chloroplast of the bundle sheath cell, that means the photorespiration has been prevented by these type of plants by this evolution. So evolution of what? Evolution of bundle sheath cell. And bundle sheath cell is revolved around my tissues, xylem tissue, phloem tissues. You will see them around this. I hope this much you are understanding. But these type of plants are known as basically C4 plants. Why C4? I tell you. Why C2? Because I was getting phosphoglycolate. Why C3? Because I was getting PGA, phosphoglyceric acid, because it has 3 carbon. So there must be one compound which must be having 4 carbons, right? We will see it. But right now you are understanding, but it has to generate glucose, right? So it has to be met with CO2. So how CO2 will enter from the surface to this particular area bundle sheet cell? So for entry for CO2 should be there. So that somebody should take out the CO2 and should reach to this particular bundle sheet cell. And over here, this C3 cycle should become so that begin so that I can get glucose so that my required product can be formed because C4 is also doing photosynthesis. In photosynthesis, I want glucose. Where is that glucose forming in C4? C4 glucose is forming in bundle sheath cell, but it is transported in a very different mechanism. In a C, and that's why we call it a C4 cycle, because the CO2 comes through this process of this particular cycle, that is C4 cycle. I hope this much is clear. First of all, just understand that this is the cross section of leaf I am trying to tell you. I have opened it up. You will see mesophyll cell. Don't try to see any other cell, mesophyll cell. Downside, they have evolved. Some plants have evolved over the period of time. They have hidden this rubisco inside bundle sheath cell so that oxygen cannot react with it and photorespiration will not happen in these plants. That means wasteful product will not be happening. right? Wastage of CO2 will not be happening, and wastage of energy will not be happening. But photosynthesis has to take place, therefore a cycle called C4 cycle works over here. So C4 cycle is basically what? Now we will see it, guys. So I suppose this is your mesophyll cell, and this is bundle sheath cell. Now you understand, guys, this is a cycle first of all. So over here, CO2 has to combine with something, right? CO2 should be combined with with someone or with something which can take this particular organic compound inside this particular bundle sheath cell where ribisco is present. And once CO2 will get to this ribisco, then they can begin this C3 cycle and they can form glucose. But CO2 has to reach somehow. So CO2 will be reacting over here in mesophyll cell with new compound, that is with new molecule, that is PEP. What is PEP? I have already told you, phosphoenol pyruvate. Now this PUP will combine with this this particular CO2 and they will be forming OAA, guys. Oxaloacetic acid. And this oxaloacetic acid is 4 carbon compound. That's why this name is a 4C4 cycle, guys, because this is the first stable product which forms over here, OAA. Right? I hope I'm clear. Now this OAA will get converted into malic acid over here in the mesophyll cell only. Mesophyll cell. You have to learn this product, guys. Malic acid will be formed. Malic acid. Now this malic acid will go into the bundle sheath cell, and this will be forming malic acid will be converting into pyruvic acid, guys. And because of this pyruvic acid, conversion of this malic acid into pyruvic acid, CO2 will be released in the process. And when CO2 released in the bundle sheath cell, it will combine with the ribisco present over here, which has been hidden from the O2, right? Then they will begin their C3 cycle. But this pyruvic acid will go again back to this particular to form PEP, phosphoenol pyruvate. And this again will react with CO2. Then CO2 will combine in the form of OAA. OAA will convert into malic acid. Malic acid will give you pyruvic acid, and they will release CO2 in the process, and this CO2 will combine with ribisco. They will go with C3 cycle, and C3 cycle will be giving you the desired product. That's why we say C4 plants are always having high yield. Why? Because they have avoided photorespiration. Never photorespiration will never happen in C4 plants, but in C3 plants, photorespiration can happen when the oxygen concentration is greater because of high temperature and high light concentration. I hope you are understanding this, guys. Also, in the water loss, if you will talk about, then C4 plants are really efficient as compared to C3 plant, because C3 plants loses more water than C4 plants, guys. So C4 plants are efficient in water efficiency also, right? I hope this much is clear, guys. I tried to explain you this particular C2 cycle, C3 cycle, C4 cycle. These words are important. You have to get familiarized. Just close the laptop, try to think, or in each cycle, after reading each cycle, try to write it down. Try to write down the words first, then their carbon number. In this way only you will be able to get it. Don't get into what is phosphoenol pyruvate, what is the formula for it. Nothing is important. This will only increase your knowledge, but time will kill, guys, because nobody will ask you about this phosphoenol pyruvate. I hope you are understanding. Your knowledge, your understanding will increase, but at the sake of this, you will lose a lot of time in understanding all this oxaloacetic acid, right? I can also explain you these formulas. I can also make these formulas, but it is not needed, right? See, another one is a very easy topic, that is CAM plants. Now, this is also evolution, right? CAM plants. Some plants, see, C3 was happening normally, but over the period of time, it was observed that C4, that photorespiration, C2 cycle began, that is basically photorespiration. So some plants evolved this C4 cycle to save them from this photorespiration wasteful product. And some plants in the areas of desert, they are not in a position that they can lose any water, right? CAM plants. Crassulacean acid metabolism. Full form of CAM plant. Metabolism. Spelling, just check it once, right? Through the PPTs. So this CAM plant is basically an evolution again. Because over here, you know that normally stomata opens at the day, but these CAM plants are found in desert areas, and desert areas, the famous plant is basically Opuntia, which you call cactus, right? So they have found one mechanism that their stomata will be closed in during the day. So during the day, their stomata will be closed. Stomata closed means they will not be able to have this CO2. If CO2 is not there, then how will they perform photosynthesis? But these CAM plants also perform photosynthesis. So a different mechanism happen over here. Same thing will happen. The CO2 is not entering. That means this mesophyll is having this particular what do we had PEP. This mesophyll cell will be having POP, and this POP will be interacting with the particular my POP will be basically stored as OAA. As I have told you, PPA will be converted into OA, and this OAA will be converted into this malic acid, and this malic acid will be stored in a vacuole over here. And then in the daytime, in the night time, this stomata opens. So they open their stomata in the night time. So opposite happens, right? So in the night time, this CO2 will enter, and this CO2 will react with this ribisco, and this CO2 will go for this C3 mechanism, and then energy will be, or say this glucose will be produced, guys. Not important, just understand what is happening in CAM cycle, how this evolution has taken place. The important thing is C3, C2, C4. This is one evolution, that is CAM. Right, guys? Again, I'll tell you once again. So if we'll see that what all we have studied till now is photosynthesis. If you will remember the equation CO2, now this equation is not just a 10th class equation. Now you know a lot of things about it, right? 12H2O gives you C6H12O6 plus, what does this give you? O2, 6 molecules plus 6 molecules of H2. Now, in order to take this, in order to have this reaction to happen, you need two processes. That means, first of all, light reaction should happen. In light reaction, we talked about basically two things. So just wait, write it down like this. In order to happen this reaction, we should go for two reactions, right? Light reaction. During this light reaction, two processes were happening, basically cyclic ETS or cyclic photophosphorylation. See, new words are there. If you will not revise it, these words will confuse you. Photophosphorylation, phosphoenol pyruvate, pyruvic acid, all this is confusing, guys. But revision will make it simpler. So don't worry if in one go you are not understanding. Take pause, rectify one concept part by part, try to understand them and rectify it. Cyclic and non-cyclic photophosphorylation. I am just writing down the words, guys. In cyclic photophosphorylation, you were concerned about FD, then you were going to cytochrome B6, then you were going to cytochrome F, then you were going to plastocyanol. I hope this is clear. Cycle you can make. In PS1 first, right? 700 nanometer. Over here, PS second, that is 680 nanometer was used. This were going to PQ plastoquinone, then it was going to cytochrome B6, then it was going to cytochrome F, then it was going to PC, then it was going completing some cycle, right? Over here, because of this particular thing, my photolysis was also happening, and because of the photolysis and because of this whole process, one thing I was getting, that is energy, chemical energy in the form of 18 ATP plus 12 and ADPH2. This was being now ready. This chemical energy now has to be utilized in my dark reaction. So dark reaction, basically the normal cycle which C3 cycle happens in every plants except C4 cycle and CAM plant. So C3 cycle is main concerned with my ribisco or RUPP. It will combine with RUPP, form PGA, phosphoglyceric acid. It will form 1,3 by PGA. It will form what? PGAL, and then it will give you fructose, then glucose, and it will go back to the cycle. So this is my C3 cycle. To in order, whenever this ribisco will have oxygen more in its surrounding, then this ribisco will combine with this particular oxygen, and this O2 cycle will give you basically what? Phosphoglycolate, and phosphoglycolate is a 2 carbon, that's why it is C2. Then this phosphoglycolate will again go ahead and become glycine, and it will lose basically CO2. It will utilize energy and will give you nothing. So CO2, to prevent CO2 cycle, C2 cycle, C4 plants came into existence. Evolution happened. Basically, evolution in the form of bundle sheath cell, and it is only about two: bundle sheath cell and mesophyll cell. Over here, C2 was all about three organelles: perioxysome, and over here, chloroplast, perioxysome, and mitochondria were involved. In C4, only mesophyll cell and bundle sheath cell are involved. C4 cell, you must remember that PEP is utilized by CO2. CO2 base combined with PEP, forming OAA. OAA is forming malic acid. Malic acid is giving you now pyruvate, and when malic acid converts into pyruvate, CO2 is released in the bundle sheath cell, and they will combine. I hope you are understanding, connecting everything. And third type or fourth type of plants are camp plants. Basically, in camp plants, what is happening? Let me tell you again what is happening in camp plants. I have told you that in camp plants, the stomata will be closed for the day. That means CO2 is not present, right? But it will open at the night. In the night, they will open. So what is happening? In the night when they will open, CO2 will enter, and CO2 is having PEP with itself. CO2 will combine with PEP. This PEP, CO2 when combined with PEP, what will happen? My oxaloacetic acid will be formed. Then it will be converted into malic acid. Then it will be converted into my pyruvate. Over here, CO2 will be released again. But this CO2 which will released and rubisco is present, it needs a light reaction to happen. But in night, camp plant doesn't get light, right? So this malic acid basically stores itself in the form of vacuole. It stored itself in the vacuole, and when in the daytime light comes, light reaction happens, it releases itself. Then this converts into pyruvate, CO2 release, ribulose, it react with this rubisco, and normal C3 cycle begins. This is my camp plant crassulation acid metabolism. Guys, this is over photosynthesis. I tried my best to explain these complicated concepts. I hope watch the video 2 3 times by part wise. First see the your light reaction, then see your dark reaction divided into C3, C2, C4, right? Like this you will be able to finish this, guys. Okay, thank you so much. I'll meet you in the next class with respiration, our new topic, right? Thank you so much, guys.